Now that we've looked at chromosomal mutations involving entire chromosomes or sets of chromosomes, now we want to look at chromosomal mutations that are the result of structural rearrangements within chromosomes. Some of these abnormalities in sections of chromosomes include duplications, deletions, inversions, and translocations. So we'll take a look at each of these briefly. Here we'll look at duplications and deletions. In a deletion, a section of a chromosome is missing. We see an example of that here on the left. If this piece of the chromosome is cut out and then the remaining chromosome is stitched back together, we have a deletion. The deletion might be due to errors during DNA replication when this chromosome made a copy of itself. It just didn't copy that section. Or it could be that the chromosome breaks, not during division, but at some other time, it breaks due to mutagens. In a duplication, a section of a chromosome is duplicated. Here we have this section of chromosome is duplicated twice and inserted over here on this side. So we have two sections of identical copies of DNA right there. So that's a duplication. Duplicated sections can all fuse to one another, like we see here, or this duplicated section could be free in the nucleus. Often deletions and duplications result in offspring that survive, but will have physical or mental abnormalities. Let's take a look at some examples of genetic diseases that are caused by deletions. One common genetic disease that we see caused by a deletion is Lejeune syndrome, or cri de chat, which stands for cry of the cat. Here we see a picture of a boy at various stages in his life with this syndrome. It's also called 5p- syndrome because of the chromosome that is affected. It's a deletion on chromosome number 5. We can see here a normal chromosome number 5, and typically with chromosomes, what we have the centromere and then we have two arms. And we typically refer to the two arms as P and Q. It is the P arm of chromosome number 5 that is affected here. This occurs in about every 15,000 to 50,000 births. It also seems to be paternal in origin, so this breaking seems to be more likely to occur in formation of sperm than it does in the formation of eggs. The second one we'll look at is DeGeorge syndrome. DeGeorge syndrome is called the 22q11 deletion, so we're looking at chromosome 22, and the Q arm of that chromosome has a deletion on it. This is the most common human chromosomal deletion syndrome, about 1 in every 4 to 6,000 births. Here we see some of the clinical features associated with this syndrome. Let's look at some examples of genetic diseases that are caused by duplications. Typically, extra copies of a gene will cause us to produce more of a particular protein, and that overproduction can lead to some developmental defects. We see a lot of neurodevelopmental genetic diseases associated with a particular section of chromosome 17. One of the common ones is the Charcot-Marie-Tooth disease. This causes abnormalities in motor and sensory neurons that supply feet, legs, hands, and arms. It affects both the motor and the sensory neurons, so we'll see not only inability to use the muscles, but also sensory deficits like cold hands and feet, tremors, nerve pain. So you can see some of the symptoms over here. We also find that duplications of some of the oncogenes, duplications within oncogenes might be a cause of some types of cancers. So we have extra copies of oncogenes, often resulting in cancer. Gene duplications also play a very important role in evolution because it appears that gene duplication was important in providing raw material for new traits that would come about through evolution. You duplicate a gene, you already have the gene performing its normal function. Now this duplicated gene can take on a new function. As these genes take on new functions, we can have a variety of different types of traits. Often when we see gene duplication, we also see the creation of what is known as gene families. You might have a variety of different genes related to one another. The globin genes are a great example. We have lots of different globins. What likely happened was there was a single gene for hemoglobin, and then there was gene duplication events, and then we end up with different forms of hemoglobin, and now we have these families of hemoglobin genes. Now let's take a look at inversions. In an inversion, a section of a chromosome comes out, flips over, and goes back in. So imagine this piece of chromosome coming out, and then we turn it on its end and put it back in. Down here we see a picture of it. Here's a normal chromosome. And now what we've done is we've taken this section, this C and D, we've flipped it over and put it back in. Now instead of B being next to C, B is now next to D. Here we've taken out and reinserted the section E and F. The difference between those two is whether or not the centromere was involved. This is representing the centromere here. It's what attaches the two arms of the chromosome to one another. It also attaches them to the centrioles during cell division. In a pericentric inversion, it involves the centromere. So it is around the centromere, and the centromere itself is involved in that inversion. In a paracentric, para means beside. It's not involving the centromere, it's beside the centromere. So here we haven't included the centromere in our inversion. It's next to the centromere. You can imagine that this pericentric inversion can change the lengths of these two arms. If this section over here that inverted was a little bit larger and it flipped over, then this arm would get bigger and this arm over here would get shorter. So if it involves the centromere, we can change the length of those arms. A paracentric is not going to change the length of the arm because this is on the same arm. It's going to flip over and go back into that arm, not going to change the length of the arm at all. It appears that a pericentric inversion on chromosome 18 had a big role to play in human evolution. If we compare our genomes with chimps, we find that we have an inversion that chimps don't have. Another important consequence of inversions is that it changes the order of the genes. If in our normal chromosome B and C were close together and they were tightly linked to one another and they were being inherited more often together because of that linkage, down here they've been split apart. Where here they were inherited more often together, now they're not. So we've taken linked genes and unlinked them. This change in gene order really affects linkage relationships between genes. If we had particular traits that were not good combinations, if B and C were bad combinations to have, and then we had this inversion, this would be a beneficial type of mutation to have because we just split up a bad combination. If B and C had been a good combination and we had this inversion, this would be a bad mutation because now we just split up what was formerly a good combination of traits. One of the things that's important when we think about inversions is to think about what chromosomes go through when they pair up. Remember, when chromosomes pair up, they're going to pair up essentially in this gene-for-gene alignment. If one chromosome undergoes an inversion, but the other homologue does not, when they get ready to pair up during synapsis, they have a problem because they're not going to align properly. If the order of this gene is now different, this top one had an inversion. 5, 6, and 7 came out, flipped over, and went back in. Now, in order for them to line up properly, this other chromosome has to form a loop so that the 5 can line up with the 5 and the 6 and the 7 can line up together. In order for one that has an inversion to line up with one that does not, we end up getting an inversion loop. So we need the inversion loop to maintain that gene alignment. However, one of the problems that occurs is that when you get this inversion loop, you don't get a crossover there. So physically, this loop is preventing us from getting a crossover. Whereas normally we would imagine being able to get a crossover almost anywhere along the length of that chromosome. Now we can't get a crossover there. What does that mean? It means then that we can't recombine those genes the way that we would have if there wasn't an inversion loop there. Sometimes they get inherited as one big package deal. If you can't get a crossover there, then 5, 6, and 7 are never going to recombine. They're always going to be in the same combination of traits. And we refer to those combinations as supergenes, where you have many genes being inherited as one big unit. Right now, because we can't get a crossover in between 5, 6, and 7, 5, 6, and 7, whatever those genes are, they're basically acting like a single gene. Here we see an example of these supergenes. In this case, the supergene that is formed is controlling wing patterning in these butterflies. One of the things that you find is that you find a lot of variety of wing pattern in these different butterflies, but you have a fixed number of different types. You don't have hundreds of different wing patterns. You maybe have 4 different wing patterns or 5 different wing patterns, and we always get sort of the same 4 or 5 wing patterns. We don't get other things. And part of that is because of the supergenes that form. Now, one of the interesting things about this is that these butterflies also have other butterflies that mimic them, and they also have the same type of supergene that controls their wing pattern formation. We'll see a video here that talks about these supergenes and wing patterns.
At first glance, it seems simple to tell one type of butterfly from the next. Their boldly coloured wings appear to differentiate one species from another. But predators must beware. The wings of butterflies tell lies. Some butterflies have evolved wings that resemble those of more poisonous species, a clever deception. In other cases, 2 equally poisonous species have evolved together to become mirror images of each other. The Amazonian butterfly Heliconius numata has evolved in this way. This one species has many different wing patterns copied from several different species from the Melinea genus. This kind of shared evolution is called Müllerian mimicry. Together, Heliconius and Melinea spread an important message to would-be predators. Don't eat us, we taste bad. Their similar appearance makes them part of a cooperative that protects them from being eaten. While scientists knew Müllerian mimicry was at work in these butterflies, it was unclear how Heliconius could derive so many different wing patterns from only one set of genes. In the mid-1970s, scientists proposed that the wing mimicry of Heliconius was under the control of something called a supergene, a long stretch of DNA that contains many smaller genes. In the supergene, individual genes are immune to the random mixing and matching that normally occurs in sexual recombination. This is how Heliconius manages to keep its various wing patterns separate. Although the concept of a supergene fits with what scientists observed in the field, no one has ever seen how it works, until now. A study of Heliconius DNA led by French scientist Mathieu Joron shows that the supergene breaks and then recombines into several different configurations. Each arrangement creates a specific wing pattern, the mirror image of a Melinea species. By mimicking its poisonous cousins, Heliconius is safer from the greedy gaze of predators, so natural selection favours these preset patterns, and by extension, the genes that create them.
Next, we have translocations. This is a type of a mutation. This is a mutation where a section of chromosome breaks off and then reattaches somewhere else to a non-homologous chromosome. It can have very serious effects or it could be benign. It just depends on how it alters the positions of the genes with respect to their regulatory region. Every gene is going to essentially have sort of an on-off switch that allows us to either use that gene or not. If I take a gene and I move it somewhere else and I don't move the on-off switch with it, then I have serious problems. I'm not going to be able to use that gene. So if I separate it from its regulatory region, then I could have some real problems. If I don't, I may not have a big problem. If it's somewhere else in the genome and I can still use it, it might be okay. In some cases of leukemia, there's a piece of chromosome 8 that translocates over to chromosome 11. We also have different types of translocations depending on where they occur. A Robertsonian translocation will essentially take a chromosome and attach it to the end of another chromosome. If this occurs, if one chromosome completely translocates to another, we just created aneuploidy because we just took two chromosomes and made one chromosome out of them. A reciprocal translocation is where we have non-homologous chromosomes exchanging sections. We have a break here that goes over to this chromosome, and then a reciprocal break here that comes over to this chromosome. So it's not the same as crossing over, because crossing over would be between homologous pairs. This is between non-homologous chromosomes. Here, there's, again, overall not any loss of genetic material. They have both of those sections. Whether or not it becomes a problem for the cell depends on whether or not we also lost the regulatory region for that particular section. So if there was a section right up here that controlled access to these genes and told us how to use those genes, then we might have a problem. But if the regulatory region came with the translocation, then probably not. That ends our section on mutation